Optical lithography is the workhorse of semiconductor manufacturing. Unfortunately, the smallest feature size that can be printed using this approach is constrained to half of the wavelength of light applied, also known as the diffraction limit. Here at the University of Utah, the Laboratory for Optical Nanotechnologies is working hard to develop an innovative technique called patterning via optical saturable transformations, or POST. It is a method that can essentially overcome this wavelength limitation and achieve subdiffraction limited patterns. Chemical dissolution is the first of two methods developed to achieve patterning via optical saturable transformations. The process begins by thermally evaporating a thin layer of diurethane onto a silicon substrate. This photochromic molecule acts as the pattern recording material. Illuminating the sample with a short wavelength of UV light converts all diurethane molecules into the closed ring isomer. When exposed to a standing wave, all molecules are then transformed to the open ring isomer, except for those at the nodes of the sinusoid. Due to the difference in chemical properties between the open ring and closed ring isomers, the closed ring isomers can be dissolved away using a polar solvent. This leaves behind a negative tone resist with features far smaller than the diffraction limit. The remaining molecules are converted back to the closed ring isomer, and the sample can be translated to prepare it for further exposures and dissolutions. The final result is a complex array of non-diffraction limited nanoscale features. An electrochemical approach is the second technique employed in POST. In this process, a layer of platinum is added in between the silicon substrate and diurethane layer to serve as a working electrode. After the substrate is illuminated with short periods of UV and visible light to create the closed ring isomer nanoscale lines, a counterelectrode is connected to the working electrode. A voltage is applied across the circuit and closed ring isomers become oxidized. Further patterning can occur before the oxidized portions are dissolved away using the chemical dissolution method. When the sample is exposed to a standing wave, the area of open ring isomers spreads outward from the peak of the wave to the nodes. The area of closed ring isomers can therefore be made arbitrarily small by increasing the exposure time. Our hope with this project is to develop a technique that is simple, reliable, and relatively inexpensive. This will help to expand the functionality and feasibility of nanoscale products.